Many thanks for the opportunity to talk about this topic. I'm not a paleontologist, but as a chemist, I'm interested in this area, which I started reading the papers about 15 years ago. Dry-aged bones, soft tissue in dinosaurs. And in this picture, we see a skull from a T-Rex, and T-Rex was the star of the papers which Mary Schweitzer, American paleontologist, was doing research on. And at least at the beginning of her research, she did also research on other topics, on other objects. And in general, if a living being dies, all the structural information decays in time, at least in an ecosystem which is well balanced. All the structural information will gone within a special time. And whenever we find fossils, remains of former living beings, there should something happen which is not aware, which we are not aware of in a well-balanced ecosystem. And so here, this T-Rex died, someone somewhere in the United States, and sedimentation occurs, and during the time, weathering will bring the object at the surface, and people can excave these bones, the remains of these bones. These are not bones, but minerals have formed these structures. Not these structures, they reset the original bones. And when Mary Schweitzer did some research at the end of the last century, she was working in a group which was headed by the famous Jack Horner. He's very famous in the research of dinosaurs. She gets some pieces which are not needed for the museum, and she was looking what does these fossils present. And surprisingly, she found in the fossilized bones, she found some soft tissue. And soft tissue not in the meaning that minerals remain the former structure of tissue, but original tissue. And if she did some demineralization in the lab of the former bones, of the mineralized bones, the fossilized bones, she also got branched vessels. And if she has done the demineralization completely, she also found cells, specialized cells in the bone, osteocytes. And really interesting for myself as a chemist, she also found biomolecular pieces of biomolecules, biopolymers. And I'll give you an introduction in her research from Mary Schweitzer, and I'll discuss some questions about. This is the area in Montana, northwestern USA, and the formations where it's very famous for digging out dinosaur fossils. And this especially is the locality where they dig out the T-Rex fossils, which was the topic, the object of the first famous publication from Mary Schweitzer. That means the first publication which the public was interested in. It's a special locality. If you want to take the fossils into the lab, you have to use the helicopter, as you see in this picture. And this is nearer to the object. Here you have the femur of a T-Rex, and this was prepared in the lab. And on this, Mary Schweitzer was doing some demineralization experiments. That means she take a sample, put some acid inside to dissolve the minerals, and she was looking for, is there something left? And it was very surprising. She found tissue in the marrow cavity of the femur. And this tissue is a special tissue we know today from birds. If birds are in the egg-laying cycle, they are suffering from calcium, and they build up a secondary bone tissue as a source for calcium for being able to lay eggs with it for the eggshell. And so this is the first. This was the first time a paleontologist was able to say this bone is from a female, from a young female, young because of the age of the bone, a huge female which is on the in an islet cycle, and this tissue is in the inner cavity of the femur bone, and this femur is this tissue is flexible. You can stretch this tissue. You can it's dehydrate and rehydrate, and it's flexible again. And if you take, and then in the way of their research, she is doing also demineralization of bone material, really hard bone material, which is fossilized. That means the bone material is reset by minerals. And she demineralized also bone material, former bone material. And she got, for surprising for all, she got vessels, connected vessel structures. In the microscope, you can see these channels. These vessels are branched, and inside the vessels there are visible some red particles in the right size for example, hemoglobin, for red blood cells. Here you have a small part. And for comparison, she used a fresh bone from an ostrich, and the same procedure, and you get a pretty similar network out of the bone if you demineralize that. This paper was harshly criticized because really nobody in the field was aware didn't expect things like these flexible tissue, vessels, or anything else. And there are critics. This may be something like a biofilm. It maybe some things of microorganisms and materials say they release. But Mary Schweitzer was going on to do their research, and in 2007 she published a second paper. She is showing some molecular analysis. We will come to that point in a few minutes. But in 2009, she published a second fossil from a duckbill dinosaur, and this is older. The T-Rex has a radiometric age of around 68 million years. This one is the T-Rex was found in this formation, Hell Crack formation, up in the upper right, and this new fossil was found 7 meters below the Judith River formation, and the radiometric age was given by around 80 million radiometric years. This is the femur, and they excaved it very carefully with a matrix around it and prepared it in the laboratory under sterile conditions. And here they are taking their samples from the femur of this duckbill dinosaur, and they could repeat the results. That means they also found tissue, flexible tissue, and vessels, and they're able to isolate single cells, osteocytes. That means special cells which are in the bones. And here they did some experiments with immunohistochemistry. That means they're preparing antibodies to actin, to two structural proteins, here actin and tubulin. And they tried these, they tried to do analytical work here in osteocytes from modern ostrich bones, isolated from ostrich bones or alligator bones. But here, these are the analytics on osteocytes from the T-Rex sample, actin and tubulin, and here from the duckbill dinosaur, also actin and tubulin. And what you see is there is a signal, a fluorescent signal from the antibody inside the cell, and you see it's not the whole cell is giving a signal, but there are only lines. That means a structure inside the cell which will give a signal from a cell which seems to be 68 or here 80 million years old. The signal is not as strong as on recent material, but it's a very clear signal from an antibody which is fluorescence marked. It's visible clearly. These are the control experiments. That means it's not an unspecific signal. What we'll see here, there are control experiments made. Further interesting is they did some dyeing experiments with dyeing on DNA, also on the osteocytes, and they used antibodies against DNA here from T-Rex, the duckbill dinosaur, and an ostrich. And you see here there is a dyeing, a stain for DNA with propydium iodide, or here with DAPI. With DAPI, it's the blue color, which indicates there are structures where dyes for DNA react and give a specific signal in something which may like a nucleus in the cell. That means there is evidence for not only for structural proteins but also for some DNA structures. That doesn't mean that there is sequenceable DNA inside, but the dyes recognize some structures to bind on. Last year, Mary Schweitzer and co-workers are doing it with another fossil from a nestling, also in the Mesozoic age, and this time not bone but calcified cartilage from a Hypacrosaurus. And here is a preparation in paraffin under the microscope, and here is staining with Alkyan blue, which marked glucosaminoglycans. And you see there is a lot of in the cartilage and little in the bones. This is a fresh sample from an emu bone, a recent emu bone, and this is the fossil. And again, you can go into immunohistochemical staining, and you will find collagen, a protein which give a signal in the fossil, a collagen 2 antibody. It's prepared by chicken and the emu also. And if you prepare the sample with a collagenase, that means an enzyme which breaks down the collagen, you can see in the fossil as well as in the emu that the signal decrease. That means the enzyme, the collagenase 2, is reacting on these collagen 2. And to show specificity, if you use another collagen antibody for collagen 1, this didn't work, whether in the fossil or in the emu bone. They also in the paper last year have indications for DNA. And now let's come to the proteins, to the molecules, to the bio-macromolecules, which are also preserved in these fossils. The main protein Mary Schweitzer is working on, collagen, is building up fiber bundles in our connecting tissues. And if you are going into the details, you find the microfibrils, and here you can, for the first time, imagine the molecular structures. This is the tropocollagen, which is a build-up from a triple helix of this protein collagen. And in our body, we have around 30% by weight of all proteins is collagen. And there is a typical sequence motif for this collagen. You have proline, the amino acid proline, and a modified proline, which is added where a hydroxyl group is added, and glycine. This is a typical sequence motif in collagen. Here I want to show in short how it's built up. It's a fantastic mechanism in the fibroplasts. They translate it from the messenger RNA the protein string, the amino acid polymer, and at the C end, there is a structure which enables the 3 protein chains to build up this triple helix. And if this is prepared in the viproplast, enzymes cut these parts here, and you have the collagen triple helix structure, which will build then the microfibers and the bundles of the microfibers. They are cross-linked by a special amino acid, by lysine, by a modified lysine, and this gives these elastic tissue character. This collagen was the target where Mary Schweitzer wants to show it's not an artifact, that we have cells, that we have amino acid signals, antibody signals from inside of the cell. And so she tried to find evidence for collagen, and she did collaboration with a researcher at the Harvard Medical School. He is doing mass spectrometry. And in short, I want to show this is a method which enables to get signals from proteins by separation the mass or fragments of the mass of peptides. And in 2007, Mary Schweitzer published the analysis of the femur of the T-Rex we saw already, and she compared it with the results of another bone from an elephant-like animal, which is aged to 500,000 years. And here you can see there are mass spectra from particles, from parts of, from peptides, from collagen. That means parts of these triple helix. Here is a detail. The mastodon, that's the elephant-like creature. They isolated some fragments and analyzed it by mass spectrometry and get these sequences. And this is what they get in the spectrum. And to prove this, they synthesized this peptide and got a very similar spectrum. And the same they did also for the T-Rex, and in a similar way. Mary Schweitzer, a quote from her publication in 2007, the presence of original molecular component is not predicted for fossils older than 1 million years. And the discovery of collagen in this well-preserved dinosaur supports the use of actualistic conditions to formulate molecular degradation rates and models, rather than relying on theoretical or experimental extrapolations derived from conditions that do not occur in nature. Because there was harsh critics about her findings, because all our experience in laboratory shows you will never ever get proteins or peptides from a fossil from such an age. And she told, they are there. We have analytical evidence. They're there, and we have to look for explanations. In 2009, she did the same also from the Duckbill dinosaur, and she got also a lot of peptide sequences she measured by mass spectrometry in the same way here a comparison from the Duckbill dinosaur and from the and compared to the same sequence in the peptide sequence in the ostrich version, and here the comparison with the synthetic version. And so she gives evidence for the existence of peptide sequences in a very old fossil from the Mesozoic age. In this paper in 2009, she quoted the hypothesis that endogenous proteins can persist across geological time, as first reported for T-Rex, was met with appropriate skepticism. However, the inclusion of additional sequence data from extant reptiles and Duckbill dinosaurs strengthens the hypothesis that the molecular signal is preserved at least to the late Cretaceous. The most parsimonious explanation thus far unfalsified is that original molecules persist in some Cretaceous dinosaur fossils. Still unknown is the chemistry behind such preservation. That means there is evidence for the existence of these peptides, and they belong to specific to tissue which we imagine in these bone parts, but we have no explanation, and the chemistry of preservation we don't know. One of the critics was that people did some research on, these are established paleontologists, molecular paleontologists, and they showed if you use samples which are C14 dated and then to calculate the kinetics, then you can show the sample from the mastodon, which is aged 500,000 years, may be possible if you look at this curve where 1% should be left from the original collagen structure. This is the age and this scale, and here we have the effective temperature. That means if the temperature during the deposition of the fossil, if it's very cold, you have a better possibility for preservation. But this calculation is excluding the existence of collagen remains in the dinosaur fossils. Mary Schweitzer later on showed, in collaboration with other people, that the parts, the fragments of collagen she found and described was inside the fibrils, and therefore maybe they are protected. And also these parts she analyzed from the fossils are at positions where today we know the collagen has functional units. These seems to be how, we don't know how, but they seems to be preserved in a special way. Here you can see this is a model of a fibril of a rat, and in the colors you can see the parts which are analyzed by mass spectrometry, and these are inside the fibrils, and therefore may be some protection. These are all indications to give an idea how they could be preserved for such a long time. In 2017, Mary Schweitzer has repeated research the fossil showed already with the developed workflow, and she showed that she could repeat with the same samples, she could repeat the results with a better amount, and she had to use smaller amounts, sample amounts, and she also found new collagen fragments and could repeat old results. Not all, but some of the former results. In this quote, she also pointed that this stabilizes and made the evidence more stronger than we really have indications for tissue, soft tissue in fossils, and also molecular preservation, although we don't know the molecular mechanism. In 2018, another working group was giving the idea maybe the Maillard reaction may help for conservation, because some Maillard reaction you may know from baking bread. The crust of the bread is the result of Maillard reaction. That means complex reactions, building up polymers in the bread and give the brown crust of the bread. And similar reactions, these researchers assume to help to protect some tissue and molecular structures in the fossils. But there is no there are no results from the laboratory to establish these ideas. Mary Schweitzer herself, she said, maybe some iron ions may help to conserve, and she did some in 2014 some model reactions in the lab and showed iron ions coming from the hemoglobin may help to conserve the tissue and also the molecule. She got some effect, but it's not enough for what we should have for the conservation of the fossils. You have to keep in mind that all the bones and the fossils very different, belonging to the location where you find them, and also in the same location we have very differentiated conservation of the parts, and also in a single bone you have different locations of conservation of things we talked about. The critics was, I told already in the beginning, there were maybe contamination in the lab when you're doing the analysis, and this is really a hard thing. Whenever you're working on samples like this, contamination is a possibility, and you have to make a lot of control experiments to lower the possibility of contamination. And the other critics is maybe there is a microbiome inside the fossils which will give you the organic molecules which you don't looking for. What I want, the point here, Mary Schweitzer is in her one of her last papers, she is giving steps we should use when we are going on to doing research on fossils like this, to grow the evidence for survival of soft tissue and protein, maybe it all at least DNA. But the points I like to underline is, when I read about the research of Mary Schweitzer, I was very surprised about her character for a long distance running, because in the beginning she published a result nobody expected. Everybody was surprised, and the main the colleagues in the field are their main critics, and she was standing alone. And I think it's really a positive thing. She listened to the critics, and she invited the critics to give ideas for testing her results and to harden the evidence. Another point in her research for me is she is arguing and working very hard for establishing her results. Nobody will believe in the beginning. But now when she is looking for explanations, how are we able to understand the survival of these tissues, of these molecules? She told, don't know the mechanism. This is fact today. There are some ideas, but we don't know. But she told the only way where she is looking for explanation is there should be a mechanism we don't know yet. And I think there is another direction where we can look for an explanation for this unexpected reservation of this material if we are compressing time. I wouldn't say these experiments will prove the time has to be shortened, but it's an idea to looking for explanations. And therefore I think this is a point which reminds us to be open when we are looking for explanations, and not to close the eyes if we for ideas for explanation we don't like. And I know some people who are doing research, who are going on in the field of creation research, and they use these results and take it for this is the reason. Here we have the proof for a short time, and I think this is not the case. But I don't understand. Of course, I understand Mary Schweitzer. She wouldn't war at all in all directions. But what I'm criticizing in her looking for an explanation is to keep out the possibility of compression of time. And this really is an idea which may lower the problems. So this research for me is really impressive, and I want to share these experiences with you. Thank you for attention.